They are amazing. Yes. Only thing you're I downloaded watching YouTube video that
He'll do your part either.
well, I just watched it. I started watching it last night. Sorry. I do that. I have to work late and watch something midnight on the vlog on my laptop, like on the side. It's still open and it's like 3 videos later. So, sorry. Oh, so when I download it to my computer, I don't know why, but I'm like not tired. Yeah. I feel like that was just maybe this weekend. I was like, is it this long in the semester after the terms where you just yeah, that might be.
Okay, does anybody have any questions before we get started? I know some people were, I don't know about you guys, but in one of my other classes, they were asking if we, before they even finished the lab exam, they're asking if we were going to go over the lab exam afterwards. And we can go over it if you guys want to. It is going to be one of those situations where you're going to see the answer and it's going to, if you like missed it, you're going to be like, okay, yeah, duh. So we can go over it if you want to. But, I mean, what do you mean? Yeah. Okay, fair.
I think I got 92.
I don't remember. I mean, you guys did well. In case you didn't read my announcement about it, my usual average on the first lab exam is usually lower than all of my exams ever. Like it's usually my lowest scoring exam for the whole semester. And you guys, this class got a 75 average. The overall average for all of my sections together combined was a 77. This is, you know, leaps and bounds better than, like, nobody has ever done that before. I want to be clear. Always have people, you know, come in just as unprepared for that as they are for the lecture exams, and then, you know, just shit all over it basically. And then again, just like lecture exams, we go over it and everybody's like, oh, of course. But I feel like with the study sheets, that's what helped everybody kind of focus on what to study. And it didn't have really anything different than the sheets that were on, like, you know, the ones that we worked on, but it was all in that one place. It was a nice little pretty, it was easier to look at. It was easier to see the information. It was more fun to look at it and all of that stuff. And I think that that helped a ton, but it proves something that I was concerned about, which is specifically that this has everything to do with quality of notes, as opposed to, clearly it has nothing to do with your ability to learn the material. It has everything to do with your ability to take notes. That's what my point is here. That's not a big surprise at all, right? We know typically when people come to my class and they're saying that it's difficult and they can't keep up with it and whatever, they are overdoing things. They're reading the textbook, they're rewatching the lectures, and there's overdoing things, not knowing how to pick out what is the important stuff and writing down things they already know and wasting their time with that or whatever it is. So, I mean, that I feel like is one of those things where with the lab study sheet, again, all I did was copy and paste stuff out of those paper. In case you didn't notice, I didn't really write much on there. I just copied and pasted what was already on those sheets. Literally just copied and pasted it in there. So, but yeah, I just picked out the stuff. I was like, okay, well, this is just describing this. We'll just put like the straight up, you know, this directly in here. And I had some pictures, but it's all in one nice, pretty sheet for that lab exercise. To me, this stuff and making notes like that, or making the ones like I went over with you guys on whatever it was, Monday or whatever, where I, you know, had, you know, here's what I do for each step. To me, that stuff is like just second nature. It's something that I do very easily. And I don't know the best way of helping you guys find that for yourselves. But we're going to try. And so my first review, my weekly review, if you haven't looked at it, and I'm sure a lot of people haven't, and that's okay. It starts off with the usual weekly review crap. But afterwards, I made notes from a lecture. It was Dr. Morford's A&P lectures with chapter 13 over the endocrine system, which I know nothing about. I never have taken physiology or anatomy ever. Okay. So to me, it's science related. So at least, I'd had like a little bit of a primer to know how to read it. And of course, I would hope that, that would be the case for people going into that in general. But like, there was stuff in there. If you watch the video, there's stuff in there that I have never heard of. I don't know any of this. And so I went through and I made notes for what I would consider would be before a lecture. So you're not trying to get everything down. You're trying to get a gist down and an organization going. And if you see something you absolutely know you've never heard of before, make a note of that, whatever. Because I have students as well who, when they, are in my class and I'm talking about stuff, they find it impossible to keep up whenever they don't know anything that I'm talking about from the get-go. So if you're one of those people that like you come in here, you don't know what I'm talking about from like the minute that I start lecturing, and you're just having a hard time even keeping up after that, maybe go through the notes beforehand, just peruse it, get something organized, like a little outline, which is really all I was doing. I had like 3 pages by the time I was done. That was it. And I reorganized it at the end. You guys didn't see that part because I'd already like stopped recording. Because a student of mine had like a question about her study practice, like personal business question. And so I just like cut out that rest of that video. But it's not any different than the first part of it. So just going through and making an outline of the stuff so you know what to expect in the lecture. So when the lecture starts, all you have to do is make notes out to the side of like the more detailed stuff. So if you're one of those people having a hard time keeping up, that's good for you. And I know that's not for everybody, but that's what the first video was about. Okay, the first of my note-taking videos. My next one on this coming Friday, so that we're all clear and we're all on the same page, so you can know if it's something worth your time. I'm still going to do my weekly most missed concepts first, and then I'm going to jump into making note sheets for at least one of these 2 chapters, chapter 15 and or 16. And I'm going to show you what the notes would look like if I was making them after the fact. And I'm going to try to make like a summary note sheet for covering both of them, because chapters 15 and 16 really, they won't seem like it at first. But once you see me going over 16, because I usually have like a review that I'll do specifically for these 2 chapters so you can understand the immune system. But they go together a lot more than you will think that they will. So they rely very heavily on one another. So we will I'll probably try to make a summary sheet. That's probably the more important thing. I feel like the summary sheets and how like the topics relate to one another are more helpful for the overall big picture stuff anyways. So that's probably what I'll do on Friday, in case you're interested. I also have a review that I held in the past for chapters 15 and 16 that I'm going to post a recording of that. I did it like, I think it was in the spring that I did it. Might have been last fall, but anyways, I'm going to post that as well. And that recording is pretty helpful. It's me literally walking through on whiteboard, kind of like you guys saw me do on the whiteboard on that like in-person thing that I ended up recording and posting for anybody anyways. But where like we talk it through, like I walk through how an immune response would work overall, like any immune response. And I make it like a chart, if you will, like a brain map of how that would work. So I'll post that too, so that you guys can see kind of how it all works together. But I feel like the goal for all of this, because I'm not going to be providing study sheets like I did. Like I have the first chapter that we went over that I went in detail, and then I had that nice pretty study sheet at the end and my notes. That's all I'm going to give you guys is that one chapter. So you guys need to learn you have to learn how to make your own notes. And if you think I am kidding, I am not when I say it gets harder from here. It does. Talk to anybody in nursing school and come back and tell me I'm wrong. Everybody in nursing school will tell you the first semester is the hardest that they've ever been through. And like it is, like when I posted on that announcement, literally the day before, I had students in like our adjunct office that come and like help us with like dissections and everything. And a lot of them are like actually nursing students. They're just trying to get, work study hours or whatever. And they're in there saying about how they were outside of that first room for the first semester students. And they were just like, they're like, I remember when I was in there and the older nurses were in there and they were outside and they were just like laughing at us. Like people would come out crying. And I'm not kidding. This is not an exaggeration. People come out of their first exam from their first semester from nursing school, often crying, upset, beaten down, all this. And the other nurses are like, yep, we told you. And they just think it's hilarious because they don't believe us when we tell you that it is bad. So that's why I'm saying, like, how about not that? How about we go in prepared and we break that cycle? And you guys come out of that test and you're like, what? Because I did it right. I did it good. I had good notes. So I'm not crying and I'm not gonna, because I know I passed that test. That's all I want for you guys. The tests when you get into nursing school, if you're going into nursing school, the tests that you're going to see at nursing school are not going to be, there's a right answer. You're going to pick the ones that are the most right. They will all be right answers. I am not kidding. You will have to pick the ones that are most right. And you will not, it's like a pass and fail thing. So if you're not prepared for that, you need to start getting prepared before you go into that. And the reason I'm focusing on it so much is because this is usually the end of the line for people before they go into nursing school. This is your last chance to get your shit together. So I want to do what I can to help you with that. Because if you don't remember microbiology, at this point, whatever with that, I mean, yeah, you have to pass the class and that's fine. But like, if you don't remember anything from this, but you at least take good note taking with you, that's what I want to leave you with a skill that you can use. And that's what I want to help you guys with. So if you find that there's something that you think you need help with, and because it's hard for me to focus on, like, if I talk to some of you guys who need help, what exactly I need to be doing to help you, it's hard for me to figure out because I don't know you as well as you do. But as we're going through this and I'm making these kind of broad suggestions and you realize, okay, well, I'm having a really hard time picking out like the general, like important topics or how to pare these things down or whatever, then you need to come let me know that ahead of time. Let me pull this up before we get too far into this. Where's the Unit 3 study sheet? There it is. Because what I would recommend is, especially as we're going through each chapter, pulling up your study sheet along. Now I can do it on my iPad, just both of them at the same time. But if you need to, if you're on like your laptop and you can have them next to each other, that's great. But if you're using paper, or if you just like having the paper versions of it, I recommend having a printed out version of it while we're going through the lecture and actually following through when we're lecturing, find the topics that are relating to what we're talking about and marking them off if you understand it. If you've made notes, you need to be making notes that make sense to you about the topics that are on the study sheet. And then circling ones that you don't understand, coming back to them and then reassessing. So that's how I would start with organizing what we're going over, because obviously there's more stuff on the slides than there is on the study sheet. The stuff that's on the slides outside of what literally is written on the study sheet is there to help you understand the stuff on the study sheet. You don't have to know every single little detail on these slides that's ever up there. Because I will post plenty of stuff up here that you don't need to know, that people I feel like are wasting a lot of time writing notes about. Okay, so let's move on. We're going to start with the innate immune system. There's two parts to your immune system. The innate just means like innately, like it does the thing on its own. It's not specific, but it's always there, kind of serving as that sort of stuff. I always see the innate immune system as like the general sort of the soldiers that are out there that are not the specialists, but they're there to fight any of the enemies, however they can, whatever they can do. But then if they come up and they see, if you're like in World War II, and you see, a German Panzer tank rolling down the street and you don't have like, a rocket launcher or something, because they probably didn't, to take it out, or sticky grenade or something like that to, explode the tracks or whatever, or you see a whole line of freaking tanks coming. What are you going to do when you're just 5 guys in your group, right? What do? You call the specialists. That's what you do. So you would radio up and say, we're going to need, bombs dropped at XYZ location, and then they'll come in and do whatever. And they still do that crap today, right? You call in the UAVs and then they will come and look at situations. Then they'll call in someone to drop bombs and whatever, or they'll launch missiles themselves from the UAV. So we still do stuff like that. But these guys are the guys that are like on the ground, getting the general view of things, doing what they can to fight the big fight. That's the innate response. The specialists that are really going to be the ones taking out the bad guys en masse and ending the war, that's going to be chapter 16, our adaptive immunity. That's where you get antibodies and stuff like that. So we're going to look at this little chart here. We have our major components of the host's defenses. Our first line being just things that you may not think of as your defense system, right? This is like your skin, because it's actually like if I take your skin off of your body, if I peel your skin off your body right now, and leave even just part of your body, like let's deglove your hand or something. Obviously, your hand's not going to be protected anymore. Your skin is doing a lot more to protect you than we realize, because we don't really think about it that way, but it is. It's our protection. It's our first line of defense. Physical barriers and chemical barriers like that. We're going to get into some details of what I mean by those, but it could be the physical barrier like the skin. It says the microbiota, that creates a barrier, right? We take up the stadium seats so the bad guys can't. And then the chemical barriers, that could be as simple as or obvious as the acid in your stomach. You know, when you're eating food, do we think that the only bacteria that are ever going to get into your body could be the ones that make you sick? Of course not. There's a lots of them that get into your body that don't make it past the stomach because they die in the stomach acid. That's actually a barrier. So then we have our second line of defense, which is the most, like the biggest focus of this chapter is the second line. And this is going to be the innate immunity proper. Things like your neutrophils, things like mast cells, and then your inflammatory response, complement, things like that. You might not think about it, but the fever is part of your response against an infection to help protect you from the infection, not just some bad side effect of it. So before we get into that, little update, okay? Listen. I talk about meatball and spaghetti all the time. And then people will be like, we know you have that your third dog. Who is your third dog again? What's the third dog? What's the third dog doing? This is my third dog, Ripley. Okay. She's old. She's a little old lady. She's like 12 now. And she's just like, I don't know. She's a sweetie. She just does not do anything particularly like crazy, like the little, little boys do. They like dig under fences and we make big ruckus and whatever. And she mostly just chills and likes to look out the window. We have a sunroom. That's where she is in this little picture up here in the middle. But yeah. And I'll tell you guys about the motorcycle too, because some people had asked about the motorcycle, but I'd started off in number 1 with the number 1 circle up the upper right. That's what I started sending my husband, because he was talking about like wanting to get a motorcycle. I want to get a motorcycle and whatever. So I started sending him things about, riding courses and like, oh, it's like a weekend. It's like 2 8-hour days basically. And you can get trained up, you get your motorcycle license and whatever. I was like, you should do it if you want a motorcycle. Well, in the number 2 down there in the bottom, couldn't fit anniversary on there. So it just says verse, but it said happy verse. You're going to need those lessons, after I'd been hounding him over the lessons. And then he was like, did you get me a motorcycle? And I did. And that's the motorcycle that I got him. And it's a Kawasaki Eliminator, for those of you who know about motorcycles. It's a pretty beginner. It doesn't even have a windshield, which he hates that like concept. I told him, I was like, if you have a full face helmet on, which you should, is what I told him, then it won't bother you if you're just driving on city streets and stuff. But because we're not planning on like going across the nation in that, it's not happening. But yeah, so we were practicing, you guys. We were practicing yesterday, and like I got it out first because I got out there first, and I was like kind of riding it up and down the street and whatever. But I grew up around motorcycles, and I used to drive a stick shift on like a car. And I don't know if you guys know this, but motorcycles are manual transmissions. If it's a motorcycle, it's a manual transmission. There are no automatic transmission motorcycles. The only automatic transmission ones are scooters. So a scooter when you see it. So anyway, so this is a motorcycle. And so I know how to shift. I just had never done it on a motorcycle before, but it's pretty, you figure it out. You get the feel for it. It's as similar as if you've ever driven a stick. It's just doing it with your feet. You shift with your feet. And so getting used to that and dealing with the clutch and all that. So I was figuring it out. And then he comes out and he's like, okay, I'm ready. And like, okay, well, remember the clutch. I'm telling him all what to do, because the clutch is with your hand. Obviously, it's completely different than a car, but you learn it, right? And he's getting it. And every time that like he kills the engine, it would like jerk forward, and he would have this panic attack and would just like freak out, and like the motorcycle would fall. And you guys, it fell, and it is heavy. And it fell like, I want to say 5 times the whole time. And I'm just, every time that it falls, I'm just like, I just bought this. Like, I didn't say it to him. I was like, it's okay, honey, this happens. And I knew that it would. It never happened to me, and it wasn't going to happen to me, but But yeah, I wasn't expecting it to be that bad. But anyways, I thought I would tell you guys about that. I'm excited about it. I'm probably gonna ride it before he does, but that's okay. I used to drive a scooter like in Philadelphia, and it was a big one. It was like a two-seater with like a trunk and stuff. And so it was a big scooter. So I know I know that. I rode on back my dad's motorcycle. He taught me about the shifting. I just never done it. So of course I'm gonna pick it up faster than he is. So I'm trying not to hurt his feelings. Like, just don't feel bad when I pick it up before you do. And he was like trying to pull it into the driveway. He's like, I'm just gonna walk it into the driveway. I was like, just get off the bike and let me drive it. Just get off. Yeah, but it was okay. He'll get there. All right, so immunology, let's get into this. So immunology, this is essentially surveying the body, finding our foreign material, and then attacking it appropriately, surveillance, recognition, and then destruction, ultimately. To recognize pathogens, basically what we call pathogens are disease-causing microbes. In order to recognize these disease-causing microbes, you have to be able to tell the difference between self and non-self, right? You have to be able to know that self we don't attack, and then non-self or foreign things we're likely to be able to attack if we wanted to, right? So antigens, we've already learned this concept, or you can refer these to as markers, any markers on the surface of the cell as the pathogen, anyways, any marker that helps you recognize it as non-self, anything on there. Usually it's going to be carbohydrates or proteins, but it can be anything else. We have already talked about how metals can sometimes be seen that way. All right. So of course you want to be able to tell the difference between self and non-self, because if you can't, even just a little bit, if there's even one circumstance where your body thinks that self is foreign, then you can develop something like an autoimmune disorder. This would include things like rheumatoid arthritis. We're going to talk about how that works in chapter 17 more specifically. Literally, like once you guys learn more about the antibodies and how they work and everything, we'll get into when things go wrong with the immune system. All right, so in the innate immune response, the most important, like as I would say, factors as far as recognizing non-self are going to be our PAMPs. You do need to know PAMPs. And this is one that I will give you like choices that are all like PAMP choices, and you'll have to choose the right one. Sort of like we had with the ELISA on the lab exam. They were all ELISA, but you had to know which one was right. So these are pathogen-associated molecular patterns. So you can already tell if we take a step back and look at that term. These are things that are associated literally with pathogens, things that cause disease, things that we would know would be bad for being in your body at all. The molecular patterns that we find on these guys. This could be stuff as obvious as, endotoxin, remember lipopolysaccharide that's found on all gram-negative bacteria. If you see that, you should automatically be thinking bad guy. It's basic stuff. Or peptidoglycan. Should we have that in our bloodstream? Absolutely not. Your blood should be sterile. You shouldn't have any bacteria in there. So if there's any form of peptidoglycan, lipopolysaccharide, and there's other ones that I think we'll mention, double-stranded RNA, because all of the functioning RNAs in our body typically are single-stranded. Viruses, though, they can have double-stranded RNA genomes. So if you see that, automatically this doesn't belong here. So these obvious are just general patterns, right? You don't associate that with specifically the flu or specifically the plague or whatever. They are very general concept of peptidoglycan, bacteria does not belong here, and we react to that. So you can get to seeing when I tell you that the innate response is very general response, this is what we're talking about. So we start very general. And we have to train our immune system at first to be able to recognize self and how not to react to self versus these like ones that we have to be able to recognize as foreign. All right, so let's get into some of the cells and tissues involved in the immune system. Start with the MPS, which isn't going to come up a ton, but it is always there. It's the mononuclear phagocyte system. That's what that MPS stands for. But it's basically just like a highway or a means by which our immune cells can migrate from tissue to tissue or between cells or whatever like that. So it's our area between there that we can use to move around. Other areas involved in all this, of course, is going to be the interstitium, like the fluid that's around that our cells can move around in as well. And then the lymphatic system. The lymphatic system is probably one that people are like understand the absolute least. Even people who take anatomy and come in here, I feel like they still don't understand the function of the lymphatic system and like what it's doing and why and how. So the lymphatic system is just like an auxiliary to the circulatory system that returns fluid from the tissues back into the circulatory system. And that's it. But in the middle of all that, we also have filters going on. And the things that you're going to go through as far as filtering on the way back into the circulatory system, lymph nodes and other lymphatic tissues like that are going to essentially be there as checkpoints. It's like if you're driving through Europe and you cross different borders and you have to go through like checkpoints, you show them your passport and whatever and get it stamped and everything like that. It's not a big old ordeal over in Europe. You can literally like, they just check it and stamp it and you go. But that's fine. But that's what it's sort of like, is like you have to pass through these filters and get checked and be sure that like everything coming through there is okay as it passes through. And that's sort of what's going on there. There's a lot of cells there to check that stuff. So lymphatic fluid itself is basically the plasma portion of your blood. Essentially, that's what it is. It might have a different level of certain electrolytes and stuff like that, but that's not what's important. It's not blood. Lymph is not blood, is my point here. It's like the fluid portion of the blood that went into the tissues. And why is it going into the tissues? That's a whole other deal that we're going to get to as well. But you have to get it back into the circulatory system. Otherwise, that can become a problem. You'll have buildup of fluid in the areas. There's more to this, I know, but we're going to get to it, I promise. So first of all, your lymphatic vessels are the vessels that are going to carry this. They have their own vessels. This is not part of the circulatory system. It's just bringing things back into the circulatory system to rejoin it. So the lymph is only going to move from one direction, from the extremities back to the circulatory system, to the heart, okay? So that's the only direction it's gonna move. And it's only gonna be pumped via skeletal muscles. Now, I'm telling you that the lymph carries fluid from extremities. I'm telling you that it's also going to be, I guess I should say this, because I hate that it starts with this, but it is what it is. So the whole reason the fluid got into the extremities to begin with is because you had some sort of injury or illness, inflammation of some kind. Ever stub your toe and realize that it'll like swell up, you'll get redness, it'll be hot, it'll swell. That's all your inflammatory response, which is innate immunity. And that's part of your immune system. And what happens is your body will receive signals because of what's going on to send fluid into the area, but not just the fluid, the cells that come with it. Things like your neutrophils and all that are going to get called to the area of injury, and they're going to move the only way they can move, which is with the fluid. So if we're sending our immune cells to go deal with, you step on a nail and you want your immune cells to go there to deal with any invading bacteria, well, they have to be pushed there with the fluid. And so they come with the fluid. That's how the fluid gets there. That's really the only reason the fluid is there, is because the cells need to go there, and cells need, their nutrients and stuff with them. So anyways, you get all these cells in this area and you get your little swollen foot from stubbing your toe on your coffee table and all that. But now it's done and everybody's happy and we don't need it to be swollen anymore. So over the next couple of hours later, as your swelling goes down, what's happening is the swelling isn't just like dissipating into the ether. Where is it going? It's the lymph. It's going to go back into the lymphatic system. All that fluid is going to move back out into the lymphatic system and drain back into your circulatory system. In the meantime, while it's doing that, get filtered by your lymphatic tissues where we have our immune responses, right? So that's good because we had to go out there to deal with that swelling. And now before you can rejoin the circulatory system, you got to go through the checkpoints. So makes sense, right? So we're building up to this concept. It makes sort of sense. The problem is that that whole movement of the fluid out of like from your toe back into your circulatory system only is driven by the contraction of your skeletal muscles. This is why if you have surgery, if you're able to ambulate, if you're able to walk, they want you to walk as soon as possible. This is also why if you can't walk as soon as possible, they'll put those little squeezy things on your legs. They like squeeze your legs alternately to try to help pump your lymph so you can heal faster because you need your immune system in order to heal. So this is the thing. All right. This is my little drawing that is very accurate for sure. And you can see like little, it's so bad, but you can see my little heart here in the middle. This guy here he is. And then we have the blood taking out, it's normal blood the cells will migrate out from the blood, which we can see in this depiction here. The cells and the fluid out from the blood into your toe or whatever. And then when it's done, it will come back through the lymph to rejoin the circulatory system, which is what this is showing here via skeletal muscle movement. Okay, so this is a picture of your circulatory system and your lymphatic system. And I don't know what this is really supposed to tell you, because to me, when I look at these two compared to one another, I don't know really, it just looks like a mess to me, but but here's the thing, right? It's hard to see lymphatic vessels in tissue. They're a lot more delicate. They don't have that sort of rigidity that you can see the thicker blood vessels and things like that. So they're not as maintained, structurally, but we have so much of these vessels going out all over our body. They are there. It's just, we don't talk about them a lot or see them a lot, anything like that. So this is a better picture of what I was just saying. It's yeah, way better, but it's showing the same thing. But you will have lymph nodes, lymphatic tissue, and vessels all over your body all the time. Okay. All right, our lymphoid organs. We have our primary lymphatic organs, the red bone marrow and the thymus. You might not, maybe if you haven't taken anatomy, you may not know that, but that the bone marrow is considered a lymphatic organ. Then you might be saying like, okay, well, yeah, a lot of stem cells come from there. All of the cells involved in your circulatory and your immune system are going to come from the red bone marrow. Yes, so those are hematopoietic stem cells is what they're called. But the red blood cells are just one. And most of the rest of the types of cells that are coming out of there are your white blood cells. And there's a lot of different ones too. In the thymus, this is an organ that is specifically for T lymphocyte maturation. So I'm just gonna put T cells, whatever, because we're gonna talk about that more in chapter, well, I guess we're talking a little bit here, but talk about more in chapter 16, especially. But yes, and then our secondary lymphatic organs, things that are, I would say, secondary to this, they serve essentially as our stations, our buildings where our workers are going to wait and see other people's passports or whatever. The lymph nodes, the spleen, and our lymphoid tissues. This is salt, malt, and galt, skin associated lymphoid tissue, mucosa associated lymphoid tissue, and the gut associated lymphoid tissue. Of course, in our gut area, we have lymph nodes, but we also have just generalized lymphoid tissue as well, and it serves a similar purpose as the lymph nodes do. So that's what these are talking about. They're not necessarily lymph nodes, but they have similar function. Maybe you didn't realize the spleen was part of your immune system. Obviously, its primary purpose, the spleen, is to filter out dead red blood cells, but it also does stuff with while it's doing that, might as well filter out the rest of the blood, is what it's basically saying. Okay, so all blood cells, all of them, red or otherwise, all originate in the bone marrow. B lymphocytes, the B ones, the ones that make the antibodies, they are going to complete their maturation in the bone marrow. However, the T cells are not, they're going to do the maturation. They're going to move from the bone marrow into the thymus and then mature there. You don't need to worry about what that means right now. I promise that we will talk about that in chapter 16, but that's where they're going to do all their maturation and develop their specificity. It's also where we're going to remove any of the self-reacting guys. All right, so the lymph nodes themselves are just going to be there to help filter out materials and provide cells that are associated with the immune reaction, because a lot of them will just hang out in the lymph nodes, like B cells. They'll just hang out there until they see something interesting. But yeah, we know where they hang out. We know they're in your groin, in your neck, and your armpit. Not need you to tell me that. So this is just a picture depicting a lymph node. When you have lymph node enlargement, this is usually because we have replication of many lymphocyte clones during an immune response. Your body will eventually see something that it definitely identifies as foreign, has a specific antigen marker. That B cell has the antibody that it can make for that. And so we will multiply out that B cell, clone it literally, that one B cell that reacts to that thing. So we can just crank out as much antibody as possible. When they're doing all that multiplying, it causes your lymph nodes to swell. So if you have lymph node enlargement, if it's generalized, just have a whole bunch of them that are dealing with that. It could have a systemic illness, but of course, more commonly what we're used to seeing is individual or local sort of lymph node enlargement, like around your neck or something like that. And that's usually localized infections. All right, the spleen obviously is a filter, but it's going to filter the blood instead of the lymph. Makes sense because it's already filtering out the blood cells. But in the immune system, it also will have macrophages there that will help eat up bad guys that it finds in the meantime. So it's like, hey, might as well, right? That's what it does. These are our associated lymphoid tissues, like we said, salt, malt, and galt. Yeah, I don't know what else to say about it, but they have the same sort of tissue there as lymph nodes have. It's just not in lymph node form. All right, blood. If you take a tube from someone, I was just talking about my husband about this because he's a phlebotomist, right? And he works for OBI, and they use like massive needles. They use they say they're 16 gauge, they're 14 gauge,
Yeah.
and so they're 16 gauge bore. And then the whole needle itself is about 14 gauge, but they don't tell people that because they don't want you to freak out when I tell you. Just for clarification's sake, this nose ring that I have, this is 16 gauge. So he was like, yeah, you know, we use a 16 gauge. I'm like, you guys, nope, this is 16 gauge. That is not, I was like pointing and I was like, that is not, that is at least 14, if not larger. And the smaller the number, the larger, if you guys didn't know that. So just to put it in perspective, the needles that typically are used with a vacutainer system that you're going to switch out the tubes on, like you can go to DLO and whatever, those are 20 gauge, so much smaller. Those are 20 gauge needles. And then when you start, butterfly needles are 22, so very small. So you will notice the difference when they're up next to each other, believe me. And the reason we brought it up is because they use chlorhexidine to prep their arms, right? They don't use alcohol. If you go to DLO and get your blood taken, it's alcohol. They don't care. It's like little needle. It's going to be in there for 2 minutes, whatever, right? But when you go donate blood, it's chlorhexidine. They don't play around. So the reason is because they're using a freaking 14 gauge hose in your arm. It's going to sit there for 5 to 15 minutes, and they don't want you to be, you know, exposed to something like that. So I was telling him about that. I was teaching everybody about like that stuff, but he's like, yeah, I don't understand, you know, something about phlebotomy. And I was like, I told him he's been at OBI for forever. And I was at OBI and then I went to DLO. And so it's a completely different experience sticking sick people as compared to, because I would say I worked at DLO, I worked at Southwest for their lab. And so their lab people do phlebotomy and they do the lab stuff. And so we had actual sick people like from the hospital coming up there. Like it wasn't like a regular DLO, like whatever. So we had sick people and we would have to stick them with the butterfly needles in their hands and all these things. I was like, it's not the same, man. You don't understand. Like they get the little needles. I was like, it's not the same. Believe me. Like it's not. It must be nice sticking people who volunteer to, you know, have their arms stabbed. People don't want to be there, especially babies. When I would have to stick babies. So you have to have somebody come out there. It takes 2 people. You have somebody come out there to hold like the arm straight. Like they'll hold it like up here and down here. And then you're just hoping they're holding it tight enough while you go for that little baby's veins. It sucks. But anyways, you know, if they bring the baby in there, that there's a reason that it's in there. Anyways, look, let's move on because I worked in the lab, so I know about these differences. But plasma versus serum. If you're going to let like plain old blood just sit there and clot, and you can then spin it in a centrifuge and it'll separate out the solids from the liquids. That liquid is the serum. However, if you have anything in there to prohibit the clotting, if you have a green top tube, a blue top tube, a yellow top tube, whatever it is, those guys have stuff added to them to keep the clotting from happening. You can still spin them in the centrifuge and you may or may not have the little wax that separates it out. But if whether or not, it doesn't matter, you have stuff in there to keep the clotting from happening, and then you get plasma. That's the difference. That's really the only difference. You get different stuff when you prevent the clotting as opposed to when you let the clotting happen. Okay, so we talk about serology. You guys have ever heard of that term, serology? Serology is the immune system stuff. It's like immune related. It's like ELISA, okay, immune related testing, serology. So serology, immune related, serum. That's really where it comes from. Serology, serum, using serum for it. So all of your immune testing is going to be done with serum, not with plasma. Okay, typically. All right, blood cells. You're going to make any of your blood cells, all of your blood cells, red, white, whatever, via hematopoiesis, and they come from stem cells. Just they could essentially turn into any kind of blood cell. They just need the right kind of signals to do so. So those are stem cells. We'll start with our white blood cells. The white blood cells are called leukocytes. That's another name for white blood cells, just a term, because science. And why not? We have two different forms of the leukocytes, the granulocytes and the agranulocytes. And it's stupid why they're separated this way, but it ends up being useful anyways. So whatever. But you take somebody's CBC sample, right? They came in, you got your little pink top tube, send it up to the lab for the CBC or whatever, hematocrit even or whatever. But no matter what, you can go stain those cells from that sample. And the white blood cells that stain with the dark granules in them are called granulocytes. And the ones that have the big nucleus and no obvious granules are called agranulocytes. So whether you're looking for granules or not in the stain, that's all it is. So it's not anything like crazy going on yet. Nothing weird. And in fact, I don't necessarily even ever need you to know what the granules really are. They're chemicals that the cells are going to use for signaling, but beyond that, doesn't really matter. Just know whether it has granules or not. Okay, so that's it. That's how we're going to divide them. And here are our two categories, the agranulocytes versus the granulocytes. All right, as it turns out. Our granulocytes over here on the right side, we have neutrophils, basophils, eosinophils, and then mast cells. So literally in that box, it says phils and mast cells. I'm probably not going to ask you about the mast cells and whether they're granulocytes or not. We will talk about the mast cells and the role they play in chapter 17, but I'm not probably ever going to put them in the context of whether they're granulocytes or not, because for me, it's easy for me to say if it's a phil, granulocyte, all of the phils, granulocytes, okay. The agranulocytes, we have some bigger differences here. We have the lymphocytes, which we haven't really learned too much about yet in the class, but this will be your B cells and your T cells, okay? Those are the only lymphocytes that we care about. Then we also have the monocytes. Monocytes, again, mono, because they have one big old obvious nucleus, big obvious nucleus. And when they move into tissues to go do jobs, they become macrophages or dendritic cells, specialized versions of those guys. Okay, so they're the same kind of, essentially the same thing, the macrophages and the dendritic cells, and they're just differentiated monocytes. So really all I've told you for the agranulocytes, lymphocytes, and the monocytes and their derivatives. It's not too bad. That's all you need to know. Red blood cells are not leukocytes. They're not white blood cells. Red blood cells are red blood cells. They're erythrocytes. And then we have the platelets, which are technically not even cells. They're like parts of cells that come off of like cells. Anyways, that's a whole other thing. That's not my class, okay? All right, the MPS, that monocyte, phagocyte, mononuclear phagocytic system is going to allow our cells that we just talked about that were up here, our granulocytes and crap, they're going to be able to move around thanks to this between tissues or injury sites or whatever. It's just their like little highway they can take. All right, so our monocytes, they give rise to our macrophages and dendritic cells. We told you that already. They are agranulocytes. They're going to live near portals of entry, essentially waiting to attack, waiting for the signal to come help. So they just chill. So monocytes are in the bloodstream themselves. Monocytes are. Macrophages and dendritic cells, once they bond to their tissues, they hang out in those tissues like forever, for always. So we have the alveolar macrophages, for example, even in their names, you know, alveoli are the little sacs in your lungs that help with gas exchange. Those guys, well, alveolar macrophages, obviously, they're hanging out there. So it's not so bad to imagine how that would work. But next, we have this concept of the cytokines. There's 4 different functions our cytokines can have, but up front, I'm just going to say cytokines, just a fancy word for our chemical signals between cells. So this cell sees a PAMP. It's hanging out in the skin. It sees a PAMP and it recognizes, oh, this is a foreign thing. So it's going to poop out all of these chemicals into its extracellular area, right? It's going to just start spitting out chemicals. And these are the cytokines. And those are going to call for this crap to happen. So in response to having seen, a PAMP or some sort of signal from tissue saying that there's injury going on, cells will produce pro-inflammatory, anti-inflammatory, vasodilators or vasoconstrictors, and then growth factors, depending on what's going on in the immune response, right? So of course, at first, pro-inflammatory. We want to get an inflammatory response going, bring in all of the cells that are involved in fighting the good fight, and that leads to inflammation. When we're kind of done and we don't need that much fighting anymore, anti-inflammatory response. That's when we start seeing your swelling go down because we have these signals telling it to go down, letting the fluid get back into the bloodstream and all that. And the vasodilators and vasoconstrictors obviously have to play a role in that too, because you have to either widen or shrink down your blood vessels in order to control what's going on in that area. You might want to vasodilate to allow blood to get to an area, but in some cases, in the case of certain kinds of infections or certain injuries, you want to vasoconstrict during the height of the fight because you don't want the thing getting out of the area. It's a way of containing it. So it just depends on what's going on and what cells call for you to do what. And then growth factors to help call for the lymphocytes to start doing their thing. The lymphocytes are B and T cells. They take a very long time to respond and then populate and grow and clone themselves and everything like that. So these guys, our innate system has to be there first. It has to be able to do something right away before we can get that lymphocyte response going. It is exactly like if you were at war and you see all these tanks coming and you're like, we can't let them get far. So let's do what we can to keep them from going until, the planes come to bomb them. Because they're not going to be here immediately when we call them, right? That's not how real life works. That's exactly how the immune response works as well. So here's some examples of cytokines that you guys don't have to memorize, but if you're interested in knowing about them, there's things like histamine that you probably have heard of that are going to be made by your mast cells and basophils that are going to lead to blood vessel, sensory nerves, and neutrophils doing their thing, their part of the reaction, whatever. So it's just examples. Anyways, let's get to the primary defenses. We already said the skin. We also have keratin in our skin. This is a term that you do need to know for the exam. Keratin is a waterproofing protein in your skin. We know keratin is in your hair too, right? It's same sort of function as well. Your hair, believe it or not, actually can act as an immune defense as well. Not as much for your head, but definitely if you think about the hairs in your nasal cavity, they're blocking things from getting into your body. It's part of your defense. But whatever, there's keratin, the hair follicles and the skin glands can also help. Sweat can help push out microbes from, your glands. Mucous membranes, and these are things that you might not necessarily think of. They're not so totally, that's not their purpose necessarily, right? We might think of sweating as being like, okay, it's a cooling effect though, right? But it also happens to help with physical barrier stuff as well. We have our mucous membranes. They're moist and they're permeable, which is a problem. Microbes like that too, right? So we don't have a keratinized layer, but that moisture, that constant movement of mucus and moisture and fluid and stuff usually involved in that, or the shedding of the mucus and all of this can help prevent microbes from taking hold as well. Blinking, tear production in the eye can help get rid of microbes in the eye area. We were talking about the nasal hairs, and in the respiratory tree down deeper down into your lungs, you have cilia that are constantly moving mucus and stuff out of the lungs. All right, the genitourinary tract, we have urine that's going to always be pushing microbes out, even higher up where you aren't controlling it necessarily. It's always going to be flowing into the bladder. Then vaginal secretions, of course, are going to help keep microbes at bay as well if they can't get purchased because of the fluids and stuff like that in the area. Right, resident microbiome. I've said this so many times, I don't think I need to go over it much more, but the deal with it. It's going to be microbial antagonism. That's our little term that we had to know about that. But that is the good guys taking up the stadium seat, so the bad guys can't sit down. They also can eliminate nutrients in that area and produce things to help deter the bad guys as well. Another term that we need to be aware of is lysozyme. I don't know why I didn't highlight it, but whatever. Here's lysozyme. What lysozyme does, it hydrolyzes. That means just breaks down. You guys remember hydrolases? Those were the guys that broke stuff apart. And then the ligases was the opposite version of it that helps join things together. So we're going to hydrolyze peptidoglycan. Lysozyme is going to be an enzyme that breaks down peptidoglycan. Pretty neat little enzyme there. Useful, pretty useful. Okay, other non-specific chemical defenses like the stomach acid and intestinal like bile. Semen has antimicrobial properties, and then the vagina has acidic pH. So these all play parts in it. I mean, I guess a lot of people may not be aware of some of these things, like the fact that semen has antimicrobial chemicals in it, but it needs all the chances it can get. Pretty much is how it, we can look at it too. So this was just looking at our first line defenses that we have just gone over and the ones that were important for us as far as how that works. This stuff pretty straightforward. All right, so we've learned about what phagocytosis is. It's whenever a cell will eat another cell. And phagocytosis, basically we're just, again, surveilling things, finding them, ingesting them, and eliminating them, right? But they also are going to help us recognize antigens later on. Some of them will. So they can play a bigger role in the overall response as we go. Our three main types of phagocytes, the three main ones, there's other ones that can help. The main ones are going to be neutrophils, monocytes, macrophages. So monocytes in general, before they differentiate. So these are our main phagocytes. These are the guys that have, I don't know why it does that sometimes. These are the guys that have a big impact on going around and eating up basically injured cells, dead matter. You see somebody get injured, step on a nail or whatever, right? The tissue in the area, because of its damage, releases just stuff from the cells that aren't supposed to be out in the outside, or create their own chemicals that are signaling that there's damage in the area. And then cells that are immediately in that area will start reacting and sending out chemicals of their own. Those chemicals call in neutrophils and eventually monocytes and stuff. And then they start doing their work and eating things up. Great. Then these guys are the big cleaning up crew, essentially. So these are like, if you had like a guy who was just a scout, that's the guys in the tissue that are always there. So the scout sees that there's the bad guys coming in, right? He's like, okay, I'm gonna call radios for help, whatever. And then the neutrophils, the macrophages, whatever, come in to help like start just mowing down everybody. But they're like, we can't contain this by ourselves. We need to call in the specialists. They call in the specialists, but they have to maintain that situation until the specialists get there. Then they can just bomb the place, just completely take it out. So that's how this is going to go. We're not going to get to the bombers today. So neutrophils and eosinophils. So we said neutrophils. They're one of our big daddy phagocytes, okay? They're really important in the early inflammatory response. So they will just come and they eat things up and they digest it, and then they go on and live their lives. They literally would just come and eat up dead cells, even and poop out whatever byproducts that it has that it doesn't need. But that's going to be important later. They're the primary component of pus, and they're actually about easily the number one white blood cell in your bloodstream. So somewhere around 50-60% of your blood, your white blood cells should be neutrophils. The eosinophils, they have a similar function, but they're usually very specific to parasitic infections. But you will see, I probably may have talked to doctors about, if you've ever had a CBC, my eosinophil count is always like really high. Always over. It's in the high. My doctor's always like, this is not high. It says it's high. I realize, but it's not actually. And I used to be worried about it, and I'm not so much anymore since I learned better. But it's allergies typically can cause them to be raised as well. So that'll come back to when we learn about the hygiene hypothesis and how we keep things a little cleaner than how human bodies were evolved to be, and how we're not exposed to things like parasitic infections anymore. And our bodies don't know, our immune system doesn't know what to do with that. It was never trained to handle eosinophils correctly because it never saw parasites. And so now we have allergies. But whatever. Monocytes, macrophages, monocytes will transform into macrophages or dendritic cells when they get into the tissue. That's really all, I don't know what to say about it. But these guys usually are going to come in and scavenge. So they come a little later than the neutrophils. Neutrophils come in just kind of right away, and they just start eating things up. and just crapping out all of the waste from eating things up. They're not super useful later on, but whatever they poop out, the monocytes can come and scavenge and clean it up. Macrophages as well. What is that? Why is that important? Because they'll pick up this stuff and carry it around and be like, this doesn't look right. And then just wave it around at the lymphocytes in a little bit. I'll go find a lymphocyte and be like, you guys recognize this? And go do that, which is incredibly important, because if we're just picking up crap all over the place and we don't know what's important and what's not, you need to be hanging around being like, okay, is this important? Is this important? Is this important? Because eventually you're going to find something where a T cell is going to recognize this and say, I know this controller. That's the controller I react to, and that's the antigen that's going to stimulate that response. If you're having an infection in your body and you can't find a T cell or a B cell that can recognize whatever is associated with that antigen, for whatever reason, I don't know how that would be, but if it did happen, then you can't have a B cell or a T cell response. So you have to have somebody going around picking up the crap, or even because they'll go in and they'll eat bacteria and stuff too and break them down. But the neutrophils were there first, really mowing it down. So these guys are just going and picking up the waste so they can find what's important and find the right T cell to activate to stimulate the immune response, the lymphocyte response. Otherwise, so see it this way, the macrophages are the ones that are holding the radio to call in the specialists. Nobody else can call them in. The macrophages can. So once the macrophages can call in, then the lymphocytes can get started on, multiplying and cloning and all of this. But man, the macrophages are probably the heavy lifters of the immune response, because they can go in there and do what the neutrophils are doing, clean up what the neutrophils did, and then stimulate the lymphocyte response, and then continue to fight with them, because they will also provide support to them. They don't stop working after that. Here is the steps of phagocytosis. You do need to know the steps in the order. We start with chemotaxis, ingestion, phagolysosome formation is not that bad, I promise, and then destruction and excretion. So this is anybody that is going through phagocytosis will do these things. Chemotaxis is just when you receive chemical signals to come do your job. Okay, so you receive a signal to the area, and we learned about this with bacteria, that they can respond to a chemical or to light, phototaxis or chemotaxis. So our cells can react to chemicals in the bloodstream and then come into the area to create the inflammatory response. Examples of PAMPs that can lead to these responses include peptidoglycan, LPS, and double-stranded RNA, which we talked about already. But so their phagocytes come in, they recognize the PAMPs. That just helps them know that it's not self. And then they will go and eat up that thing. So, hey, I recognize this, peptidoglycan, recognize it, and then just eat it up. And that's all they need to do for now. Next, they form the phagolysosome. Well, when I ate up that PAMP, so controller, I recognize that this is the controller PAMP on it, and I eat it up. Well, that vesicle that I formed around it is called a phagosome. Zome, and it's a vesicle in general, okay? So we've created a vesicle, phagosome, and we're going to merge it with a lysosome. Boy, if that doesn't sound familiar, it should. Lysosome, right? So in fact, it does have lysozyme in it, as well as a bunch of other toxic things. We're going to have free radicals like our toxic oxygen molecules, and maybe some acids and things like that. Just a whole bunch of stuff that's toxic in that lysosome. And it's going to merge with the phagosome and then cause it to die, break it down, kill it, and everything like that. Death to the bacteria within 30 minutes of that happening in your cells right now in your body, probably. So yeah, when they're merged, they become the phagolysosome. That's just the word for it. All right, moving on to inflammation. We have 4 major Latin-based words that are associated with the inflammatory response. Rubor is redness, calor is warmth, tumor is swelling, and dolor is pain. I don't speak any Spanish, and that's a shame, but I'm sure if you speak Spanish, some of this is a little easier for you. But for those who may not, at least the ones that I can tell you, I mean, rubor, obviously, you can see that would be red like a ruby. Tumor, obviously, swelling like tumors would be. And calor, if you think of calories, that actually means the heat that's given off by, burning, a carbohydrate or something like that. But that's what calorie is referring to, is the heat that's given off. It's the heat energy. So, hey, warmth, that makes sense. And then dolor, I just need to know what the word means. It's easier if you speak a language based on Latin. We have another sign too. It's a fifth sign, and it doesn't get a Latin word for some reason, but it means loss of function. That's going to be our loss of function aspect. So typically when I'm going to ask you about the signs of inflammation, I'm mostly going to be talking about the words up here, rubor, calor, tumor, and dolor. So these loss of function guys, it more or less is going to happen as a result of when you start seeing excessive inflammatory response associated with something. But you can see that, right? I mean, I don't know what else to say about it. Chronic inflammation as well can lead to development of other disease presentation. If you are just constantly having the presentation of those symptoms up there, those 5 signs of inflammation, can obviously lead to what you might think is a disease, but is in fact just inflammatory response carrying on for a long period of time. It can cause damage, but it's not so much the inflammation that's causing damage. It's like the chemical signals involved in it are causing the cells involved in the response to release damaging things, and those can cause damage. All right, chief functions of inflammation. Yes, it does have a purpose. This is going to be to attract immune components, to start the repair process, to clear away any harmful substances like toxins or things like that the organism might have made, and then to destroy the microbes themselves and block further invasion into the area. A lot of times you'll hear about how immune cells will like wall off an area. So when we see, pus forming in like boils and things like that, that's all been walled off and localized to that one area. Your immune system did that for you. And yes, all these things are happening as a response. So you can see when we start talking about these aspects up here, you have rubor because of the vasodilation. The blood is flowing into that area to bring the response to that area. And the warmth happens, of course, along with that, right? The increased blood flow. Blood is warm, especially in the extremities. Your extremities are typically cooler than the rest of your body. So having increased blood flow there makes it warmer. You're going to have swelling because the fluid that's bringing the cells into the area to fight the fight. That's the swelling. We call that edema, right? And then we have the pain. That's stimulation of the nerve endings. It's just something that happens in the area, but it's also a good thing. It just lets you know that something is going wrong in that part of your body, in case you didn't already know because you stubbed your toe on your coffee table. It's just basically how it would be playing out. You step on your nail here. You have your bacteria that are going to be getting in your wound. And then we have our mast cells that are like already hanging out in that area. They start releasing chemical mediators. Now they can't eat anything. They're not part of our phagocytes, right? Remember what our phagocytes were. Those were our neutrophils and our monocytes and our macrophages mostly. So they aren't going to eat anything up. They're just saying, oh shit, okay. So they're calling in somebody to come help. They saw something, that's the scout. They saw something, they're calling in for help. The chemicals they release get into the bloodstream, and then they're going to call in the neutrophils. So here come the neutrophils. Oh, you know, we have little things here. I always forget. So our immune cells that live in our tissues respond to injury and infection by releasing our chemical mediator. Okay, cool. That's not anything different than what I said. All right, then we have the second one, the neutrophils, which just wouldn't pop up like that every time. Neutrophils arrive along with the plasma that carries them there when the immune system calls them there. So cool. So it was the mast cells, right? They called in with their little chemicals. Now, finally, the neutrophils are squeezing out of those. You see the little picture where it looks like they're literally like just squeezing out of those blood vessels? That is actually literally what they're going to do. They're going to squeeze between the cells of the blood vessels and then go into the tissue. It's crazy to think about, right? I don't know. Anyways, our next step, this one up here. Cool. Neutrophils and plasma and bacteria are going to form pus. And then I don't know what else to say about that, but yes. So you're going to have all these dead bacteria. You're going to have the neutrophils eating them up, pooping out their little products. You have the fluid that came into the area with the neutrophils, and the neutrophils themselves. All of this forms pus. And most of pus is actually neutrophils. Okay, really. We just want you to collapse, and you're just not going to compromise, are you? I just have to get it. Okay, there we go. All right. Anyways, the last one, finally. So we had the neutrophils that were doing their thing. But while they're doing that, we had sent signals out for more help, and that was for the lymphocytes. We've already called in the bombers, right? Now, finally, our lymphocytes have come in. Our macrophages are helping along with everything because they'd come in and called in help for the lymphocytes. Macrophages are a lot bigger than the lymphocytes, but they're also doing most of the work. So lymphocytes usually, the T cells and B cells are often like helping mediate this whole response. Not entirely. We'll talk about more of what they do later. And they're absolutely necessary for creating memory so that if you see this thing again, you can fight it off. Without our lymphocytes, our T cells and B cells, you would not have any memory. You just keep getting infected by the same thing every time you saw it, always. So yeah, they're there. Anyways, they usually, once they show up though, the party's over. Like they ended it. That's it. That's all you needed was for them to show up. It just takes them a while to get there. Okay, so things happen in your blood cells that I don't really care about other than, the chemokines are going to send signals and lead to letting fluid to get into your tissues and blood to flow to the area. Then we have edema, which is buildup of fluid because fluid got into those tissues, that fluid that got into the tissues and escapes outside of the blood and is around in the tissues called the exudate. So edema is buildup of exudate. So all that fluid moving into that. It can be clear or it can be pus. And that ability of those white blood cells to squeeze in between the cells of the smaller blood vessels, this is called diapedesis. And that is a term you need to know. There's going to be a question probably on the test asking about how do lymphocytes and white blood cells move from the blood vessels into the tissues. The term for that is diapedesis. It's literally just squeezing in between the cell junctions. They're able to change their shape and whatever. We already said about chemotaxis. There's certain signals that will call in certain white blood cells, and literally, like there's all sorts of different signals. This signal might call in this cell. This signal might call in that cell. This signal might call in all cells, whatever it is. And there's all these different chemicals that your body can make. And we saw some of them in the previous slide, but maybe you've heard of some of them, things like interleukin or interferon or tumor necrosis factor, or they might heard of it as just TNF alpha or whatever. You've probably heard of some of these at some point in your life, either seeing it on a commercial or whatever it is, because there's a lot of medications out there like Remicade and Humira that block the function of these guys. So now when I say that now, when we're learning about this, that makes a little bit maybe more sense. But they are literally like adalimumab, which is Humira, is literally an antibody that blocks the receptor that the inflammatory response, TNF, tumor necrosis factor, which is part of the inflammatory response, it blocks it from binding onto what it's supposed to on cells to signal the inflammatory response. It's great. So you completely blocked it from docking in where it's supposed to create that inflammatory response. But we have just learned that you need that inflammatory response. That all of this is happening for a reason. So that's why you also have increased susceptibility to infections if you decrease that response. So edema has its purpose too. It'll dilute the toxic things in the area. It'll help, brings in clotting factors, and the clot that leads to forming there helps create a trap so that the microbes can't move around. Then neutrophils will also arrive with that fluid to come fight the good fight. Long-lived inflammatory reactions. Okay, so after we've attracted our monocytes and our lymphocytes, whatever they might be, and then the macrophages will clear up all what was in the pus. That would be the actual neutrophils, the dead bacteria, all the byproducts, everything like that. So they'll start cleaning that up. And then the B lymphocytes will have antibodies to react to for the foreign molecules. The T cells can literally directly kill invaders, as well as the cells that might be infected. And then tissue will repair itself with that scar. Fever is part of our inflammatory process. It is, when we talk about the calor part of it, it's not just about that one localized area. It can happen throughout your entire body. So fever has its purpose. Of course, we know kind of the ranges of the fever. I'm not going to test you over those, but you know, just for personal reasons, you might not like to know it. Things that stimulate a fever are called pyrogens. If you think of like a pyromaniac being somebody that likes to light things on fire, think about it that way. So pyro being fire, being like hot. So fever. So if it stimulates fever, it can come from outside of the body or from inside of the body. So what do I mean by that? If it is like a microbe, an infectious agent, a chemical that's stimulating the response, that's exogenous. If it's your immune cells sending signals to have a fever, that's endogenous. So it's just saying that your body can send signals to create the fever, or there are parts of like microbes that can just stimulate a fever, like LPS. Endotoxin is very potent. So what are the benefits of fever? It inhibits multiplication of temperature-sensitive microbes, just more than we think. Impedes nutrition of the bacteria because it inhibits the availability of iron in the area. Because they need that too. It slows down enzymatic reactions for the bacteria, increases metabolism of our own body so that we can stimulate our immune reactions faster. Speeds up, like it says down here, speeds up hematopoiesis and all these other things. All right, should you treat the fever? I'm not going to tell you what to do on this. Talk to your doctors, of course, but the recommendation tends to be to let it run its course until it is at a level that you feel is unsafe and needs to be treated. You talk with your doctor about what you think that is. All right, next is interferon. Interferon is one of these chemokines, one of these, or cytokines, it doesn't matter what you call it, but it's one of those chemical signals that's really cool for your cells to basically let each other know that there's an infection going on. So it's for a viral infection, but it's not virus specific. It's for if there's any viral infection in a cell, okay? So it's a protein made by white blood cells and tissues that mediates our immune response for viral infections, all right? I don't care if there's somebody at my door. Okay, synthesis, I need to silence this. I always forget to silence it. All right, okay, so you can actually make this and infuse people with it to help them fight off infections and even cancer. It's because if your body sees the cancer as foreign, which is, we would hope we can stimulate that sort of a response, but this helps with that sort of thing too. So with a viral infection, we've got Jim and we've got Kenny, okay? Kenny's like the neighbor, or he's like down the street or something, okay? So Jim is this cell down here on the left, and he is being like getting infected with viruses. And he's like, oh no, I'm being invaded. And so he goes and hides in his closet and he says, I got to call Kenny and warn him. And so he does, and he calls Kenny and he says, thanks for the warning, Jim. I have my shotgun ready. And he's ready when the virus shows up. He's got all of these antiviral proteins already made. His shotgun is loaded and he's waiting at the door for these guys so that will prevent or at least slow infection with the virus. And that's what interferon does. It's interfering with the viral cycle. This is my best example, and I feel like it's pretty accurate. Okay, so interferon is that warning call. Lastly, we have complement, and this is the complement cascade. Complement is a set of proteins in your bloodstream, like 50 proteins or something that's always there. This doesn't have to be synthesized or activated or brought into the area. No, it's always there, okay? And they work together in a cascade reaction, which just means that you activate this one protein and then it activates the next one, and then that activates the next one, and then they activate the next ones, and whatever, and so on and so on. So that's how it will go. So it's a sequential physiological response. All right, there's 3 ways you can activate this cascade: the classical, the lectin, and the alternative pathways. The classical is activated by antibodies. So if antibody is present on the surface of a microbe, then the classical complement pathway will be activated. And these aren't cells, right? These are just proteins that are in the body, and they just activate each other when there is an antibody present on something, okay? Or there's certain sugars on microbes that this molecule lectin can bind to, and when that happens, same thing, trigger it. And then finally, alternative is just going to be repeating molecules. We'd say this is sort of like the PAMPs. It's not the exact same molecules, but similar concept. But this includes LPS. That's the biggest example. Okay, so it sees things like repeating molecules and that'll trigger it too. So antibody, sugars identified by lectin, or the repeating molecules in the alternative pathway. Okay, so here's a little picture at the top. We have the initiation of our complement pathway, either by antibody, lectin binding to the sugar, or, you know, LPS or something like that. Okay. Next, we're going to have the different steps, you know, cascade, whatever. And all these intermediates that are involved in the cascade can do different things. But there's 3 major functions of complement. One of them is going to be opsonization. Let me get a pen that is white. One of them is going to be opsonization. This is, and it says phagocytosis right next to it, right? I just want to be clear about this. But opsonization is a fancy word for basically helping out phagocytosis. And what I mean is, remember how we had the capsules, and it's hard for our phagocytes to like eat things because of the sticky and everything. Well, opsonization means we're putting these complement proteins onto the surface of that capsule and making it easier for the phagocytes to grab on. That's opsonization. It's just giving like a handle for the phagocytes. All right, so that's opsonization. Then we have inflammation. We learned about that. That's not too tricky, but it helps signal and create that response. And then we also have the membrane attack complex. And the membrane attack complex is like the biggest thing that it's going to do. All right, so we had that initiation. We have the amplification and cascade. During that, we can have opsonization and inflammation. But at the end, which is the big goal, we get the polymerization of the proteins that are, you know, all cascaded. And they come together and create a hole in the surface of the microbe and help it just leak out its contents. It's very effective. It's a membrane attack complex. So yes, just some words explaining that. Initiation, we either initiated via classical, which is antibody, lectin, which is the sugars, and then alternative, which is LPS or similar. Amplification and cascade, we're just each of these molecules triggering one another. We can also have the opsonization and inflammation. And then finally, we have the membrane attack complex formation from polymerizing or bringing together all of the parts. So we'll get holes in that membrane. And that, I believe, is that. That's the last one, yes. Okay. All right. Okay. Anybody have any questions about any of that stuff? Okay. I brought these because I thought that I had the ones with questions. So my bad that I didn't. I guess you guys aren't crying, but. Today is in lab is going to be as quick as you guys let it be. I mean, we're going to do gram stains. Some of the samples that I made for you guys, a couple people, like maybe I think one table has some samples that didn't grow very well, but everybody else should be fine. You'll still have something to work with though.
Are we doing more of the like
I was like, yeah, no, looks like there's
immune working days? The
I know.